Welcome to the presentation of our paper, Brainomaly, Unsupervised Neurologic Disease Detection Utilizing Unannotated T1-Weighted Brain MRIs. I am MD Mahfuzur from Arizona State University. This work has been done in collaboration between Arizona State University and Mayo Clinic and is funded by Department of Defense, National Institute of Health, and Amgen. It is difficult to acquire annotated samples for training anomaly detection methods. Therefore, many anomaly detection methods use only healthy data for training. However, anomalous data are usually available at training time, just unannotated. In this paper, we explore how do we utilize unannotated T1-Weighted brain MRIs to train a deep learning model and improve unsupervised neurologic disease detection. In this paper, we explore Alzheimer's disease detection and headache detection. For Alzheimer's disease detection, we collected the dataset from ADNI database, and for headache detection, the dataset was collected at Mayo Clinic. An image-to-image translation-based method in the literature solves this problem in 3 stages. First, an image-to-image translation model is trained to remove diseases from all the input MRIs. In other words, the model learns to translate any brain MRIs to MRIs of corresponding healthy-looking brains. Therefore, if the input MRI is already healthy, the image-to-image translation model is expected not to change anything. Once the model is trained, all the brain MRIs in the test set are translated to their healthy-looking brain MRIs. Then, subtracting the generated healthy brain MRI from the input brain MRI reveals diseases, if any. We call the resultant image a difference map, and the residue in the difference map helps us to detect the presence of the disease. However, this method has some limitation. We can improve the disease detection by addressing these limitations. The first limitation we will explore is in the image-to-image translation. The existing method translates the entire image. Therefore, it is possible that the healthy region may change more than it requires. For our applications on brain MRI, we observe that the brain MRIs can be registered. Therefore, we can avoid the cycle consistency loss required for unpaired image-to-image translation and predict only the voxel changes that are required to make the input brain look healthy. So, our first major contribution is improved image-to-image translation. In our proposed Brainomaly method, instead of generating the healthy image, we generate an errative map that identifies which voxels in the input MRI need to be changed by how much to make the brain look healthy. Then the final healthy brain's MRI is generated by adding an errative map with the input MRI followed by a 10H activation. We include the standard loss functions from the image-to-image literature except the cycle consistency loss and follow the standard training method. Our proposed image-to-image translation improves the unsupervised Alzheimer's disease detection by 27% and headache detection by 13%. This ablation study on the objective function shows that our choice to include the identity loss from the literature has a positive impact on our image-to-image translation. The second limitation of the existing method is in its Inference model selection. Since we do not have any annotated sample during training or validation, it is not possible to select the best performing model for inference after training. The existing method utilizes FID to select an inference model that produces the most realistic images. In our experiments, we observed that FID does not select the best performing disease detection model. Therefore, as our second major contribution, we propose a pseudo-AUC metric for inference model selection. To calculate our pseudo-AUC score at the validation stage, we first translate a set of unannotated MRIs and calculate the disease detection score by translating them into MRIs of healthy brain. As we don't know the annotation for these MRIs, we assume their ground truth is disease. Then we use another set of known healthy brains MRI to get their detection score. For this set, we already know that their ground truth is healthy. Now, combining the predictions of these two sets, we calculate the AUC score, which is the score of our pseudo-AUC metric. For inference, we select a model that produces the highest pseudo-AUC score. Our experimental results show that the proposed pseudo-AUC metric improves the disease detection score by additional 2%. Our proposed pseudo-AUC score also shows higher correlation with the real AUC score compared to the FID score. As our model utilizes unannotated MRIs during training, it can operate both in transductive and inductive setting. Our ablution study shows that our model provides statistically similar performance in both of the settings. Here are a few qualitative results. As expected, we see the brains with Alzheimer's disease or headache show higher residue in the difference maps compared to the healthy brains. So in summary, we introduced a novel unsupervised neurologic disease detection method that utilizes unannotated images from diseased and healthy subjects. We proposed a new metric, pseudo-AUC, for selecting a suitable model for inference when an annotated validation dataset is unavailable. Our detailed analysis proves the superiority of the proposed method over the existing methods. Empirically, we showed that our pseudo-AUC metric has a higher correlation with the model's underlying disease detection performance and selects a higher performing model compared to FID. Thank you.